I did my presentation this week on stem cells. So first we just have to look into what are stem cells. So stem cells are cells that have the ability to differentiate or develop into different kinds of specialized cells. So what a stem cell can do is they have two paths. They can either self-renew or they can differentiate into any kind of these specialized cells. Specialized cells range anywhere from a skin cell to a nerve cell to a heart cell to a blood cell. So whether or not a stem cell replicates or differentiates is major, right? We need stem cells to self-renew themselves, so we have a constant supply of stem cells in our tissues. But we also need them to differentiate in order to replace specialized cells that die or get damaged or used up. So having stem cells is really important, especially stem cells that can differentiate into specialized cells that aren't capable of mitosis or dividing, right? So like neurons and things like that. So there are two major types of stem cells. The first are embryonic stem cells. So embryonic stem cells develop in blastocysts. So in a lab, if you fertilize an egg after about 4 to 5 days, that will turn into a blastocyst. And what it is it's a group of embryonic stem cells surrounded by a trophectoderm, which is just like an outer layer. And what scientists can do is they can isolate the inner cell mass of all of those embryonic stem cells by removing the trophectoderm. The important thing about embryonic stem cells is that they can differentiate into any type of cell in the body, right? So there's no limitations. This is called pluripotent. So that capability is really important for research scientists. However, it's a bit controversial just by the nature of how they obtain these cells. The other type of stem cell are tissue and adult stem cells. So these are the stem cells that we find in our body that we need for things like repairing damaged tissue. These are multipotent, so stem cells that grow, or they're found in tissue, so they've been found in hearts, brains, bone marrow, things like that, but they can only differentiate into the type of cells in the tissue that they're grown with. So for instance, stem cells in red bone marrow can only differentiate into different types of blood cells, so white blood cells, red blood cells, and platelets. What scientists have recently discovered are induced pluripotent stem cells. So this was a major discovery. In 2006, scientists found that they could take a skin cell from a mouse and genetically reprogram it into a pluripotent stem cell, which means that it can differentiate into any type of cell. So it acts like an embryonic stem cell, but it's not an embryonic stem cell. So you avoid the controversy while also getting a stem cell that could be very important in personalized medicine. So the thought with that is, if you took my skin cell and I had a disease, right, and you took that skin cell and you genetically reprogrammed it into an iPS cell, any kind of tissue that you form when you differentiate it can go back into my body, and my body shouldn't reject it because it originally came from my skin cell. So this was discovered, I think they found it in mice in 2006, then they made iPS cells from human cells in 2007. So it's fairly new, and there's a lot that's not yet known about it. And the whole thing with genetic reprogramming, what they do is they introduce new genes, which are like codes for our cell. They introduce it to the cell to basically like how a computer programmer has a program, they can introduce new code to that program to make it do a different task. So that's basically what scientists do when they turn like a skin cell to an iPS cell. So how do stem cells differentiate? It's basically a combination of internal and external signals, either secreted chemically by neighboring cells, or direct cell-to-cell contact, or mechanical strain. So if you remember in the blastocyst, it's this big pool of embryonic stem cells, and eventually all those stem cells turn into, it grows into a fetus and then into like all of us today. And it does that by differentiating. So it can differentiate, you probably can't read, but there's 3 different layers: ectoderm, mesoderm, and endoderm. And so these 3 layers make up the embryo. The inner layer is the endoderm, outer layer is ectoderm, everything in between is the mesoderm. So how they get there is through chemical signals and all these different types of things. However, there's a lot of different other types of cells in our body. So to go further down, scientists have tried tracking different pathways to get it there. So if you wanted to grow a stem cell in a culture and differentiate it into anything you want, it's a lot more complicated because you need to make sure you're growing it in the right environment, exposing it to the right growth factors and chemicals, and if there are other cells in the body that help differentiate it, you need to make sure those are present too. So it's a lot harder than just kind of reprogramming it and then making an eyeball. You can't do it, like at least now. The future is stem cells. So if scientists could use stem cells and just make them whatever they want, it would have major implications in treating diseases and things like that. So currently, right, we can go from embryonic stem cells or from iPS cells. If we grow it under conditions A, we can get skin. If we grow it under conditions D, we can get liver cells. We could then build tissue with those cells, but since we don't know or aren't really aware of how those conditions need to be run, it's a little hard to do this.
Please contact the main office.
So with more research, scientists can really hone and track those pathways. Issues though are that growing stem cells in culture can be a bit difficult because cells often behave in ways that you don't expect. At Hopkins, in a laboratory at Hopkins at the Wilmer Eye Institute, they're growing iPS cells, and one of the postdocs there had been growing hers for like 6 months. But one day she went to go feed them and they were all dead, right? She didn't plan for that to happen, but you spend all this time and sometimes it goes a different way. However, if everything were to go right, the implications would be, growing new insulin-producing cells for diabetics. So if you could take an iPS cell and produce insulin-producing cells to make up for the pancreas that isn't functioning well, then implant that into a diabetic, you could theoretically cure them. Treating Alzheimer's and Parkinson's, if a part of the brain is afflicted and you can grow new neurons and better tissue and implant that, they are cured. Basically, there's no limitations, right? If you can repair any kind of damaged tissue and implant it, it could be amazing. Another thing that they're working at the Wilmer Eye Institute, one of the doctors there is trying to grow iPS cells and produce tissue of retinal pigment epithelial cells. So this is a cell lining behind the retina that helps take light and produce electrical signal that helps you see. Right? So people with age-related macular degeneration, which is very common in old people and causes blindness, right? They have a very damaged layer of these type of cells. What he wants to do is he wants to grow his own RPE cells from their own skin cells, produce the tissue, and then implant it back into them. And he hopes that this would cure them. So it's a very individualized, specialized medicine that hopefully works.
Good morning.
Because that tissue shouldn't reject
students
their
arriving.
body
We
because
wish
it
them
came from
all
their
the
own
best
skin.
today in their competition. Best of
Other
luck
issue
and
is that
bring home the trophy.
there are a lot of
Thank
risks
you.
that they want to be able to alleviate. So tumor formation, unwanted immune responses. The hope is that if you took a skin cell and produced tissue with it, or different tissue that you wanted to put back into your own body, there could be some kind of unwanted immune response, so there needs to be more research done in that area. However, I think it's certainly worth it because if scientists can make a breakthrough with these iPS cells, the implications are so vast.